So this is the last video of the content that I'm providing to you before your IB exam. This is on the last section for the SL component of the options, which you will see as part of paper 3 next week. So D4, which is all about the heart, I want to make sure that you understand that this is additional content to what you need to know about the heart in chapter 6. So don't think that you don't need to know anything about the heart for papers 1 and 2. You just don't need to know all this detail for papers 1 and 2, but you will need it for paper 3. So might as well learn it and know it now. And so the first thing I'm going to talk a little bit about regarding the heart is the cardiac muscle structure. It's labeled 1 up here. I did my best to do a drawing here, but I would definitely cross-reference with your book. So have your book open because they're pretty good diagrams in there. And I just tried to abbreviate that drawing here to simplify it to maybe make some sense to you so that you could use this kind of diagram to explain the cardiac muscle structure. So first thing to note is that it's actually quite similar to your skeletal muscles in that the protein that you find in these muscle cells is actin and myosin, which are type of proteins. And that these muscles in your heart have special structures because of their unique functions of maintaining contractions involuntarily. And so they contract without the stimulation of any nerves for your entire lifespan, which is quite incredible. And they have certain features that prevent them from tiring or fatiguing, which is also quite incredible, because if it didn't do that, you wouldn't likely survive to this age and be able to do all the other things that you do. So the structure of these cardiac muscles are kind of like a Y shape. They have branches on the cell, so they have a unique shape. It's not that lie that we tell you when you draw that animal cell as a round or spherical cell. It has a Y shape, and the idea there is that it can actually share resources with neighboring cells. There are these things in between the cells that are called intercalated disks, and basically it's a double membrane, just like any other membrane, that have gap junctions. They have interconnections between neighboring cells that allow those cells to share cytoplasm, which means that they can share ions, which allows for action potentials to be able to be sent through those neighboring cells very quickly. Unlike the nervous system that requires action potentials or electrical signals and then neurotransmission or chemical transmission and then action potentials again, this allows for continuous electrical impulse, which allows for very quick propagation of that signal down the neuron or down these cells. So the rapid movement of ions reduces the resistance. So if you are in physics, with electricity, you should know something about resistance, voltage, current. And so the idea here is that by reducing the resistance, it allows for rapid depolarization. And when you hear that word depolarization, I want you to think about the graph of action potentials and think about that peak, rising to the peak on that chart as the depolarization, when the internal membrane becomes less negative, and it allows for the propagation of that signal down the neuron. And so in the effect of all of these things is that that network of cells will contract as if it's a single cell. It's not a wave of contractions, it's contractions simultaneously, which is quite incredible that the heart, or parts of the heart, really, I'll explain this in a second, can contract as a large unit instead of single cells, like in your muscles, in your skeletal muscles. So we need to talk a little bit about how that signal gets propagated through your through your heart. And the first thing is something that's called the SA node. That's how I'm going to abbreviate it. The book talks a little bit about it as the SA node. You need to know it as the sinoatrial node. And so that's where the SA come from. It's from the sinoatrial node. And so this is where the signals originate. And really, you don't need to know how they originate here, but you should know that signal is systematically, rhythmically propagated, but it can't be passed directly from the atria to the ventricles. It's actually going to be instead passed from the SA node to another node called the AV node, or the atrioventricular node, and I'll talk about that in just a second. So what the SA node does is actually quite incredible for the cardiac cycle. So I'm going to scroll down here. So what I've done is try to simplify this cardiac cycle. There is a diagram that shows you what's happening in each of the chambers of the heart. This diagram is that schematic diagram, but the book has a better diagram of the heart that might help you see what's happening with the valves. I've just made this a bare bones diagram. So there's a series of contractions and relaxations that happen in your heart that constitute a cardiac cycle. So if we were to start measuring it at the atrial systole, which is defined up here as a contraction, so when the atria up here contract, they force the blood that's in that atria into the ventricles down below. Now these ventricles, to get to the ventricles, these AV valves, or atrioventricular valves, have to be open, and when they open, these semilunar valves, the valves that leave the heart, the valves in the vessels that leave the heart, close. And it's not so intuitive in this diagram, but again, if you look at the book, you can envision the blood flowing and what it does to the valves. So in the atrial systole, the atria contract, force the blood into the ventricles, then the ventricles go through systole, or contract, and they force blood out. When they force blood out, the AV valves shut. When they shut, the atria fill back up from the pulmonary vein and the vena cava. So after the ventricles push all their blood out through the contractions, or the ventricular systole, then everything relaxes in your heart. Everything is then relaxed before the SA node sends a signal to the atria to contract as a network of cells, which then triggers the ventricular systole. Now the way that happens is that ventricular systole, there's a very important, I'm going to put a star here, a very important delay that happens right here in the cardiac signal. And that delay is due to the SA node sending a signal to the AV node. And the AV node then continues sending that signal to what are called Purkinje fibers, which cause the ventricles to contract. And I'm going to outline this in just a moment, and how this happens, and really why the delay happens here between the SA node and the AV node, but then also how the Purkinje fibers speed up that contraction so that the whole ventricle closes, or both ventricles close as a single unit instead of a wave action. So let's go up here. So why is that delay so important? So I put a green star next to it. So why is that delay so important, given that I told you that the whole heart, or the heart, works as a network of cells? Well, really what happens is that the atria work as a network and the ventricles work as a network that contract within each network. So the fibers that connect the SA node and the AV nodes conduct that action potential slowly. So that same signal that's being sent through these Y-shaped cardiac muscle cells in the atria gets slowed down on their way to the ventricle. So the atria work as a network contracting, and then there's a delay sent to the ventricular network of cardiac muscle cells that then will contract as a separate unit. So the AV node, which is also in the right atria as well as the SA node, takes longer to get excited. So if you remember that graph again for the action potential, it has to hit a threshold value to cause the action potential, but the AV node has a greater distance to go to get to that threshold value. So the resting potential is actually even more negative than the -70 that we talked about, Therefore, it takes even more of a positive charge internally to that AV node to cause the action potential to kick off and go through that all or nothing principle, because it's got to hit that threshold value. And part of that is due to the AV node having a smaller diameter. So you'll have to trust me on this. I can definitely show you the proof of this in class when I come back. But when a nervous cell or nerve cell has a smaller diameter, the propagation velocity is slower. Given that there are no other adaptations like a myelin sheath or Schwann cells. So if there's a smaller diameter, the propagation velocity is much slower. And so the AV node has a very small diameter. It sends that signal slowly. And what you need to think about here is that having a small diameter increases the surface area to volume ratio, but it makes the propagation velocity slower. So you don't want always a slow propagation velocity. So there's a balance that has to be struck between an efficient surface area to volume ratio, but also a fast enough propagation velocity. And that balance is the beauty of where natural selection comes into play here. Because when propagation velocity is fast enough, you survive and can pass down genetic material. And you can get things in and out of your cells efficiently, you can survive to pass down genetic material. But what's good enough is dependent on the environment and what you need to be able to respond to. That's an aside. Also, the AV node has fewer sodium channels, voltage-gated sodium channels. So because of the higher or more negative resting potential and fewer sodium channels, an action potential, once reached, will actually not propagate as quickly. There's also a prolonged refractory period, and that's because there are fewer mitochondria, less ATP, therefore less active transport happening with the sodium-potassium pump that's trying to restore the resting potential before the next action potential can occur. There are also fewer gap junctions, and remember that gap junctions allow for a cytoplasm to be shared, so the signal can be sent as a network. So there are fewer of those in the AV node. So why is that delay so important? So we talk about why that delay is happening, but why is that important to know? And that's what's here in green. So the atria, so the reason that delay is so important, and the reason that the atria and the ventricles don't contract at the same moment, is that the atria can empty all their blood from the AV before the AV valves shut. So all the blood in the atria can be removed from the atria before the valve here, the AV valves, the tricuspid and bicuspid valves shut. And if they don't shut, and we don't want them to shut too quickly, because if they did, they would reduce the amount of blood in the ventricles, which would reduce the amount of blood flowing through your body. So if you think back to the video on D3 on the liver, there would be issues with nutrient regulation, there would be problems with detoxification as well, and if you think back to the hormones and homeostasis video from a few weeks back, there would also be issues with temperature homeostasis because heat is being transported through your blood vessels. And if you can't move blood fast enough, you may not be able to get rid of enough heat. And you know the effects of high heat on enzyme action. And in fact, that's true for all proteins. All proteins will be denatured. And as you guys are realizing, I hope that proteins are quite important to keeping you alive. They are to quote David Goodsell, the machinery of life. So once the signal gets through the AV node, that again is here in the right atrium, that signal has to then, after being slowly transmitted, has to actually be transmitted extremely quickly, so that both sides of the ventricles can contract at the same time. And so it has to be done very quickly. And so that's the lower network of nerves that are going to contract. And that's, again, where the Purkinje fibers, here are the Purkinje fibers, they reappear in my notes, have specific have specific adaptations. So I'm going to add, I guess what I'll do, let me grab my pen here. I'll try to do is add, so there's an SA node up here, and there's an AV node as well. Excuse me, sorry for that note there. The AV node. And so the SA node will cause a contraction of the atria, but then there's a signal that's sent to the AV node. It's slow. The AV node takes a while to pick it up, but once the AV node picks it up, it goes into the septum, or the divide here between the two cells. It divides into two sides, and then comes up on either side of the ventricle. And so these Purkinje fibers are those fibers that are coming back up, and they have specific modifications to allow for very rapid transmission of that signal, so that again it works as a network to contract. And so here's that list. They have, it's kind of the opposite list of the AV node. They have a larger diameter, so again you have to trust me, a larger diameter would increase the propagation velocity. There's an increase in the density of sodium channels, of voltage-gated sodium channels, so action potentials can be reached much more quickly. And, well, excuse me, not action potentials reach quickly, they actually will, the amount of time that it takes for an action potential to run through its entire cycle would be very much reduced, or happen much more quickly. And there are also many more mitochondria and glycogen stores which are used to reduce the time for the refractory period. So again, the glycogen would be broken down into glucose or some form of carbohydrate or energy molecule that will be transferred in the mitochondria into the ATP molecules, which will then pump the sodium-potassium pumps to restore the resting potential of the cells. And that glycogen was made from insulin. So think back to the pancreas now, from the insulin that was made in the beta cells of the pancreas when there was higher than normal blood sugar levels. So I hope that you see that this is actually connecting very well to the rest of our curriculum. The last small point to make is what causes the sounds for the heartbeat. And there are two sounds. The medical professionals often refer to these as the lub and the dub. Lub, dub, lub, dub, lub, dub. And so that lub actually is due to the AV valve snapping shut. And so this is at the start of the ventricular systole, not the atrial systole, the ventricular systole. And so when the ventricles snap shut, the ventricles contract. When the ventricles contract, the AV valves snap shut, and that causes the lub sound. And then when the blood, after the ventricular systole, the semilunar valves will shut, preventing the backflow of blood. And again, look at those diagrams in the book that help you understand a little bit why that's happening. It's not so clear in this schematic diagram. That shutting is the dub. So the AV valve shut is the lub, and the semilunar valves shutting is the dub. So it's lub dub, lub dub, lub dub, lub dub, lub dub. And so the time between the lub dubs, what's happening is we're looking back at the cardiac cycle, we've got the diastole, or the relaxation of all of these muscle fibers, and then the atrial systole. And then when the ventricular systole happens, we get our lub, and when the ventricular systole is finishing up, we've got our dub. And so that happens over and over and over and over and over again. And it's that pressure of the systole and the diastole that doctors are measuring when they take your blood pressure levels. They're looking at kind of the strength of your heart, and the tension in your arteries and veins, and determining if you have hypertension or not. Hypertension being an indicator of possible plaque or atherosclerosis, or buildup in your arteries and veins, which is an indicator, could be an indicator of high amounts of cholesterol and saturated fats in your diet, or if you're putting together some information from the video on D3 and D1, the liver making cholesterol for your body, but it not getting out of your bloodstream quite fast enough. Okay, so that's a lot of information. You may need to watch that more than once. You definitely need to process this. You definitely need to anchor this on your structure of the structure of the heart and action potentials from the nervous system. Okay, and that's a wrap.